Hello, everyone. Welcome to the Genetics 311 video for problem worksheet number 12. Today's topics are eukaryotic regulation, immunogenetics, and developmental genetics. First, let's talk a little bit about the levels of control for eukaryotic gene regulation. These are gone over in detail in your lectures, so I won't expand upon them here, but at least let's just list a few. In prokaryotes, we said that the main level of control is at the level of transcription. And that is certainly a level of control for eukaryotes as well. With eukaryotes, we also think about the alteration of chromosome structure, particularly chromatin remodeling. If you think about the nucleosome structure where the DNA is wrapped around an octamer of histones, and then remember the H1 histone, the H1 is the little yellow thing here, the DNA is the gray, and the octamer is that big circle ball in pink. The H1 histone protects the linker DNA. That octamer is not always wrapped around the same part of the DNA. You can kind of think of that sliding around a little bit. And that's important because the DNA that is not wrapped as tightly to that octamer is more likely to be transcribed than the DNA that's wrapped tightly. Also, that wrapping is not static. See this picture here? You can see that there are little tails of the histones that are holding that DNA tight to the octamer of histones, versus here the tails have been acetylated and the DNA is loosely wrapped. DNA that is loosely wrapped is more likely to be transcribed. So in general, we need to get these other levels here. After transcription occurs, in the nucleus, we have processing. So we have control at the level of processing. And this would be the processing the primary transcript of messenger RNA. Then we have transporting that processed messenger RNA out to the cytoplasm. Once that messenger RNA is in the cytoplasm, it might be translated, it might not be translated. It might just kind of hang around. So translational control at the level of translation. And then some messenger RNAs have a long half-life. They are very stable. They live for a long time. Whereas some messenger RNAs have a short half-life and get degraded rapidly. So we can think about stability of the messenger RNA. And then we can also think about protein modifications. I did not really talk about protein modifications other than to mention them in the lecture. So now let's look at this a little bit more closely. In general, transcriptionally active DNA is more open than non-active DNA. And when we think about that, that kind of goes with our tightly wrapped, not very active, versus loosely wrapped, more likely to be expressed. So when we think about histone acetylation, this is not acetylated, this is acetylated. So an increase in histone acetylation is associated with an increase in gene expression. I don't have a picture for DNA methylation here, but that was discussed in a fair amount of detail in the lecture, particularly methylation of the cytosine residues. And in general, highly methylated DNA has less gene expression than less methylation on the DNA. I wanted to briefly refer to this diagram from the lecture. This is one I drew, but this one just for a minute. This is the basal transcription apparatus where RNA polymerase is associated with our DNA, and all of these little bubbles of various colors are the various transcriptional factors. And in the book, it probably will tell you which transcription factors are which and call them by name. I'm not concerned with you knowing the specific names of the transcription factors, but I do want you to realize that they're going to complex with each other and with the DNA and help to recruit RNA polymerase so that RNA polymerase can start transcription. Another thing I wanted to point out is this little piece of the DNA in yellow up here. This is an enhancer. And I realize that the magnification here is not easy to read, so I'll write it out. An enhancer is a sequence of DNA, and it helps to promote additional transcription of genes near it when it is complex with the transcription factors and brought near to the site of transcription. So now take a look at the diagram on the left. I've kind of stretched out the whole gene. The basal transcription apparatus would be associated in this area. +1 is our start of transcription. And then we're going to transcribe this DNA. Here's the poly A signal. So eventually, once that messenger RNA is formed, it'll be clipped after it reaches the sequence transcribed from that, and the poly A tail gets added. I wanted to point out that there are two components to the promoter, a core promoter and a regulatory promoter. Core promoter is closest to the start of transcription, regulatory promoter a little further upstream, but both of these are upstream from the start of transcription. And here is my enhancer. Enhancers, as I said, are sequences of DNA that promote transcription of nearby genes. They can be upstream, which is what this one is. They can be within the gene. They can even be downstream. And they can be tens of thousands of base pairs away from the start of transcription. There's one thing an enhancer can't do. It cannot work across an insulator. So that brings us to our little problem at the bottom of the page. Here I have genes A, B, C, D, E, and F, an enhancer sequence, and an insulator. And the question is, which genes will be stimulated by this enhancer? This enhancer can work backwards and forwards, so it can stimulate genes A, B, C, and D, but it can't grow across an insulator, so it will not stimulate genes E or F. So I'm going to leave these problems for you, but I'm going to talk just a little bit about cis and trans. So a transacting versus a cis-acting. We use these terms in prokaryotic regulation as well. Remember, a transacting element could work and affect all the pieces of DNA in the cell, similar to our repressor in the lac operon. We have a similar situation with eukaryotic organisms in that there are transacting elements. Anything that is a protein that's made by a gene, if you have a bad one and you have a good one, the good one maybe can do the job that the bad one couldn't do. And that would be a transacting element. Cis-acting elements are not proteins. They're going to be segments of DNA. So our enhancer would be a cis-acting element. It's only going to be able to work to change the expression of genes that are on the same piece of DNA versus our transcription factors from the previous picture are transacting. I'm going to let you answer those two questions, and the key on Moodle will have the answers for you as well. Let's move forward and talk a bit about the immune system. Our immune system is really, really amazing. If you think about it, we have 22,000 plus or minus genes in our genome, and we can make probably an infinite potential of antibodies. Well, that's kind of puzzling considering that an antibody is a protein, and you think about a gene coding for a protein. So clearly, one gene has to be able to code for multiple proteins, particularly since the number of genes that cause antibody production is a teeny piece of that 22,000 genes. So what happens with our immune system genes so that we can make such a large number of antibodies? There are three things: the somatic recombination, junctional diversity, and hypermutation. So let's look at how these work in this particular situation. Somatic recombination is like DNA processing. It's the step that occurs going from germline DNA, such as what I have here, to get to the mature B cell. It's going to cause some chunk of this DNA to be removed so that the mature B cell has shorter DNA than the germline sequence. Well, let's take a look at this problem. In this problem, it says that ultimately this sequence in this particular cell makes an antibody that has the V2, the J3, and the constant regions. And you notice we have a lot of variable regions, we have a lot of joining regions, and the constant. So at the end of the day, we want V2, J3, and the constant region, and that's it. The first step is the somatic recombination, and that occurs in the maturation of the B cell. In this particular way, everything that is between V2 and J3, see if I can draw that to here, all that is removed. And that's removed in that somatic recombination. So now I can draw, let's see, we've got V1, V2, and then we'll have J3 hooked to that because we took all this other stuff out. So there's J3. And we still have J4 and J5 and constant region. So this is somatic recombination, where we've removed this sequence. The junctional diversity comes into the fact that if there were two different cells that were putting V2 and J3 together, that junction would be a little bit different. So maybe I would leave a little bit here or leave a little bit there. So maybe they overlapped a little more, a little less. And that's going to cause the final messenger RNA to have a slightly different sequence. The other part is hypermutation. Hypermutation only occurs in the variable region. So from the point here that at the end of the day, V2 is the only variable region in our antibodies, we would be concerned about hypermutation in the V2 area. But normally we get maybe a 1 in 100,000 is our mutation rate. The mutation rate in the variable region of these genes is 2%, which is like 2 in 100 or 1 in 50. That's a lot higher than 1 in 100,000. So a huge amount of mutation going on in these variable regions. Well, once we've got our mature B cell DNA, we have to transcribe it. And we make that primary transcript or the pre-messenger RNA. And that's going to start transcription right here and go all the way down. So effectively we have this V2J3 J4, J5, and the constant region. But at the end of the day, our messenger RNA just has to have V2, J3, and the constant. So all of this part right here is processed out. We've talked about removing introns before, and this is just processed out as removing an intron. That's different from the somatic recombination when we took out a chunk of DNA. Here, we're just processing messenger RNA, and that gives us our final product. I want to spend the rest of the time talking about developmental genetics and a couple of model organisms. So in this problem set, we're going to talk a little bit about Arabidopsis thaliana and Drosophila melanogaster. I believe in the lecture, I also mentioned a little bit about development in C. elegans. So when we were talking about the development in Arabidopsis, we were talking about flower development. And if you think of a flower, you can think of it as concentric whorls. The outermost whorl, sepals, that's the part behind the petals, usually they're green. Then you have petals. In this case, for Arabidopsis, they're white. Then stamen, Arabidopsis has 6 stamen, and then carpels in the middle. And Arabidopsis has 2 fused carpels, so it looks like one structure coming up. So we can think of those 4 whorls. And the model we looked at was the ABC model of flower development. And if you recall from lecture, we labeled A, B, and C like this. And we said that we were effectively dividing our flower into those 4 whorls, and different classes of genes worked in different whorls. Remember, whorl 1 was the sepals, whorl 2 petals, whorl 3 stamen, and whorl 4 carpels. And just a refresher, the stamen, that's the part of the flower that's going to produce the pollen. That's the male part of the flower. You can remember stamen, male. And the carpels are in the very middle. That's the female part of the flower, and that ultimately makes the seeds. In order to produce sepals, we have to have all the A genes functioning correctly. In order for petals to be produced, B and A have to function correctly. For stamen, we need B and C functioning correctly, and for carpels, we need C functioning correctly. Notice that there are some T-bars because A and C work antagonistically to each other. If they're both present, they toe the line right in the middle where they're supposed to. But if there's a C mutation, effects of A will kind of trickle over into these inner whorls. And if there's an A mutation, effects of C will trickle outward. And that's just something to remember. The other thing is that the C gene holds the number of whorls to 4. If we have a C gene mutant, sometimes we get more than 4 whorls. Okay, the problem part here comes where we're looking at these flowers and we're trying to determine which class of genes is messed up. So let's see, there we go. Now you can see the model and the flowers. Sometimes you can look at these pictures and you can see what structures are present. And sometimes you look at the pictures and you just have to go, what structures are absent? And between the two, you can figure out which gene class is messed up. So these green things are sepals, the white are petals. So sepals and petals, that means A and B are functioning correctly. This must be a C class mutant. I don't see any stamen or carpels. The other thing is I have multiple whorls. It looks like there's more than one whorl of petals there. C class mutants sometimes give you a flower in a flower, which is really kind of cool. Now let's look at this one. Well, there's a nice carpel right there. So clearly I have C functioning just fine. And these little things here are the tops of the stamen. So that means B and C are both functioning fine, but I don't have any petals. We would notice those big white petals. And also I realize it's hard to tell the difference between a sepal and a leaf. That's a leaf-like structure. It's not a real sepal. So the A gene class is messed up here. Here we're looking straight on the flower. Oops, there we go. That's a normal flower straight on. This is our mutant. I see some green sepals there. I see a little bit of green sepal behind and petals and more petals here. This is a C-class mutant. We do not have stamen or carpels, but we do have a lot of sepals and petals. Now this next mutant, if I look at it, that is a normal sepal, and that's a normal carpel. That kind of looks like a sepal, but it's not in the right place. And so it's also mutant, and there aren't any stamens showing. So this is a B-class mutant. We've got normal sepals in the outer whorl and normal carpels in the middle, but something's messed up with the center two whorls. And let's look at this last one. That's a nice big carpel. And here we have a stamen. Now this flower, we just don't see all of the parts. We clearly don't have normal sepals or petals. Looks like that's some sort of abnormal structure. So this is an A-class mutant. Now in your homework, we were looking at things a little bit differently. In your homework, we were looking at genotypes. And we're saying that one capital letter is a dominant allele, and that means that class of genes is working well. So for this particular genotype, the A-class gene is working, B-class is mutant, and the C-class is working fine too, because big C is dominant to little c. And then you were asked something about what structures were present in each of the whorls. So A-class genes working fine. That means we have normal sepals. B-class genes are not working fine. So that means whorls 2 and 3 are mutant. A plant anatomist would tell you exactly what structures those are. I wouldn't. So, and you don't need to know those either. If it's not the normal structure in the normal place, you write mutant. And then the C-class gene works fine. C by itself makes normal carpels, so we should have normal carpels. So this is more similar to the way I asked the questions, I think, on the homework. Okay. Let's move to Drosophila. So I was going to introduce just a little bit. These are a couple of the slides from the lecture. They even have the speaker. So I wanted to introduce the idea of the genes that we looked at in the early embryological development in Drosophila. We talked about 3 main classes: maternal genes, segmentation, and homeotic genes. And we broke the segmentation genes down into the gap, the pair rule, and the segment polarity genes. So in order, the genes are maternal genes, gap, pair rule, segment polarity, and homeotic genes. So that's just kind of a refresher from lecture. I encourage you to go back and watch that part of the lecture. But the idea is we start off with our developing embryo. It's going to get its poles, your anterior and posterior end, your dorsal and ventral side established, and it's going to get divided into segments. Bigger segments, then smaller segments, and then getting the anterior-posterior orientation for each segment. And with these homeotic genes, we actually get a final identity of each segment. And what's really cool is that the segments laid down in the early embryological development hold all the way to the adult. And that's even more amazing when you think that it goes from the egg through 3 larval instar stages, the pupa, and then the adult. So these structures for the adult are determined and where they're supposed to be early in embryological development. So that is just, I think, incredibly cool. One of the genes that we looked at is called the dorsal gene. It's the dorsal gene. It makes the dorsal protein. And the dorsal protein is a transcription factor that's important in determining the dorsal ventral axis of the fly. So the dorsal side, this is the dorsal side, is the back. And the ventral side is down here. And that's going to be the belly of the fly. So we got the back and the belly. The way this works in naming mutations, when you have a mutation in the dorsal gene, you get dorsal structures on the belly of the fly. So they look at the fly and they go, oh, wow, there are dorsal structures here. And they named it dorsal. Now, you and I would probably maybe not go that way, but it makes sense from the naming notation. It makes less sense when you think about how this works, because the dorsal gene makes a transcription factor that stimulates the development of ventral structures. So it's kind of a funny nomenclature. But normally, since it's a transcription factor, it's going to be in the nucleus of ventral cells, and then it means it's going to be in the cytoplasm of dorsal cells, because if it was in the nucleus, it would be making ventral structures on the back, and we don't want that. So when this protein is in the nucleus, it's a transcription factor, and it's going to regulate other genes in development to help make the proper ventral structures on the belly of the fly. So that means this dorsal protein is going to be found in the dorsal messenger RNA, is going to be found in the more belly-oriented nuclei. And if you look, I drew it better on the slide in the lecture, because there actually is a gradient. So a lot of it down here in the most ventral area, and less as you go up towards the back of the fly. Now, we also have maternal genes that are important in establishing the anterior and posterior axis. And initially, it's the bicoid, which is in the anterior side, and the nanos, which is in the posterior side. The bicoid messenger RNA is anchored up here. So bicoid here, you can sort of see it. And nanos will be on the posterior end. And we actually have a gradient of expression. And that's these genes both influence action of other genes, including hunchback and caudal. Again, from the lecture slides. So bicoid is going to regulate translation of hunchback and caudal. The hunchback and caudal messenger RNAs are pretty much all the way across the embryo, but bicoid is anchored at the anterior and nanos is anchored at the posterior. And it works such that bicoid is going to prevent the activity of the caudal gene. So it's going to prevent tail structures from forming in the anterior side where the bicoid is strong. But it's going to promote the anterior structures by activating the hunchback gene. So our little question, we've already put genes in order. I already talked about that. So here is where bicoid would be. And on the posterior side is where nanos would be found. Now, those gradients are really important. And this is a good time to talk about arrows and T-bars. When we think of an arrow, we're thinking of promoting the activity of or causing the transcription of. When we think of a T-bar, we're thinking of preventing the activity. So you'll see arrow and T-bar symbols. So normally, caudal promotes tail structures, but bicoid inhibits the expression of caudal. So this would be a way to write this developmental pathway. From the anterior point of view, and you can remember caudal and tailbone, so that kind of helps you figure that that's going to be something that's in the posterior. Think about hunchback, kind of near your head, and so that's going to be something that's going to be important with anterior structures. So hunchback normally promotes the production of anterior structures, and bicoid is going to promote the activity of hunchback. Well, hunchback is still going to promote anterior structures no matter where it is. However, if we're at this end of the fly, the nanos is anchored there. It's going to prevent the hunchback gene from functioning. And that means we will not get head structures at the tail and tail structures at the head, but hopefully we will get head structures at the head and tail structures at the tail. So that was a little bit of an introduction to developmental pathways. And our lecture has a couple of developmental pathways. I think there was one with Harry Potter, and I think there was one with the Hunger Games. So I came up with a Harry Potter one, and my daughter actually, I was trying to explain it to them. They came up with the Hunger Games. So let's take a look at The Simpsons. This one is more complex than what I would normally give you, so don't panic if this one seems a little challenging. When we think about how to figure out a developmental pathway, we look at mutants. So in this case, I have Bart, Lisa, and Marge, and Homer are the various genes on my pathway. And Homer is my end product. So the question is, does transcription of Homer occur or not occur? And I look at all sorts of various mutants. When Bart was mutant, the transcription of Homer does not occur. And that tells me that when it's functioning correctly, Bart is promoting Homer, right? The transcription of Homer. Because if it's messed up, it doesn't promote Homer, Homer doesn't transcribe. And mutants lacking Lisa, transcription of Homer does not occur, which also tells us Lisa normally promotes transcription of Homer. And then in mutants lacking Marge, transcription of Homer does occur. So if we don't have Marge, we get transcription of Homer. That means if Marge is working right, it blocks or prevents or inhibits transcription of Homer. Okay, but the problem is we still have a bunch of little pieces. We haven't put things together. So we can look at some double mutants. When I had a double mutant of Bart and Marge, transcription of Homer occurs. So I look to see which of the single mutants, Bart or Marge, causes transcription of Homer to occur. When Marge is mutant, Homer occurs. When Bart's mutant, it doesn't. That means Marge has to be closest to Homer in my overall chain. And it means Bart's back here. The question is, what is that symbol? So let's think about it. What can it be? Whoops, too big of a piece of paper. Okay, the symbol could either be an arrow or a T-bar. But whatever it is, we have to ultimately make it consistent with these two pieces of information that we already had. If Bart is mutant, Bart does not promote Marge, which means Marge cannot block Homer, which means Homer transcribes. Well, that doesn't work because in mutants lacking Bart, Homer doesn't transcribe. So that's not correct. If Bart is mutant, Bart does not block Marge, so Marge blocks Homer. So when Bart is mutant, Homer does not occur. This one's correct. So that is a T-bar. In double mutants lacking Marge and Lisa, transcription of Homer doesn't occur. So again, transcription of Homer doesn't occur. This time I'm looking at these two single mutants. This one doesn't occur, that one does occur. So Lisa must be closer to Homer than Marge is. So Lisa, and we said Lisa arrow Homer. Lisa promotes Homer activity. And we got to figure out Marge and what that symbol is. So we've got our two possibilities. If Marge is defective here, Marge does not turn on Lisa. So Lisa cannot turn on Homer. If Marge is defective here, Marge does not block Lisa, which means Lisa turns on Homer. So in a double mutant with Marge and Lisa, we have transcription not occurring, which means we need to make this a T-bar. Normally, Marge blocks Lisa. If we have the double mutant, then we are not going to get transcription because Lisa can't turn it on. But if we block Marge, Marge doesn't block Lisa, which means we do get Homer being transcribed because Lisa promotes Homer. So that works. So now we can put that together. Bart, block, Marge, block, Lisa, promotes Homer. Now, if none of these genes are mutant, everything's working just fine. Does Homer activity occur? Do we transcribe Homer? So Bart prevents Marge from working. That means Marge does not block Lisa, so Lisa does cause Homer to transcribe. And what if we lacked both Bart and Lisa? Bart is not here. It's not going to block Marge. Marge will block Lisa. Well, Lisa's broken anyway, so Lisa does not stimulate Homer, so no, that one will not happen. Like I said, this is probably the hardest of these types of problems I could ever think of, and it is far harder than what I would expect to give you on an exam or your homework. I'm going to leave this last problem for you. It is a little bit easier because here it's telling you when this protein is active, it stimulates the ventral structures in the fly. So it's telling you what arrow to put there, and then it's asking you some questions about it. Well, that's it for this rather long problem session video. Thank you for your attention. I hope that this has been helpful, and please do contact me if you have any questions. Have a great afternoon.